I'm going to speak to you about mycorrhizae at the extreme. When we say extreme conditions, extreme soil conditions, extreme environments, and so on. So first of all, what is mycorrhiza? Mycorrhiza is an obligatory symbiosis among soil-borne fungi and plant roots. From the plant taxa, we have this association with this symbiosis with almost all angiosperms, gymnosperms, gametophytes, pteridophytes, sporophytes. Almost all the photosynthesized plants, they have the ability to form Arbuscular mycorrhiza symbiosis anyway, in general. From the fungi genera are Basidiomycota, Ascomycota, and Glomeromycota. Glomeromycota is the most important phylum of fungi forming mycorrhiza symbiosis. So continue to what is mycorrhiza. Mycorrhizas and not roots are the main chief organ, or as they say, it was described by Smith and Reed, of nutrient uptake from the land plants. The study of plants without their mycorrhizas is like study an artifact, so something ideal, something which doesn't exist in nature. Because everywhere you go and everywhere you sample roots, you will find them in symbiosis with mycorrhiza symbiosis. Okay? So what it does, from the plant, the plant provides the photosynthesis, the carbohydrates, and actually feed the fungus. The fungus has the need for carbon, so the plant provides this to the fungus. In return, the fungus provides the plant with soil nutrients. That ability started with a fungus many million years ago, something like 1 billion or maybe before years, and the fungus, all these fungi, they have the higher ability from the plant to track nutrients from the soil, to absorb, to uptake from the minerals, some cations, for example. You have to spend a lot of energy. The plant doesn't have that ability, not to so high extent. So that's why the plants form this symbiosis, this mutualistic symbiosis. Beyond that, as you can see in the right picture, the fungus extends in a high degree the rhizosphere of the plant. The rhizosphere is the small zone around the roots. It's something like 2 millimeters around the natural roots of the plant. So it forms a huge area. The plant, with the association with the fungus, they can uptake nutrients, water, and everything else from the soil in a much larger area. So to the benefit of both fungus and the plant. As you can see there, all these brown lines are from the young pine tree, and all the white things are the hyphae from the fungus. So they form a unified zone, call it a mycorrhizosphere. So it's a unite thing. So why we have to look why the below ground organisms are important, and specifically more the mycorrhizas. So there's a increased interest on soil ecology and soil mechanisms as for the microorganisms. So there are some books, for example, they are ecological books. The guy who wrote, the main author of this, actually returned the image of the tree, having just trying to show the importance of the root system or below ground organisms. Below ground biodiversity is a key factor on ecosystem functioning. Mycorrhizas are one of the key factors to form the plant societies, the plant, how do you call it? Associations, you know, the plant communities. And now there's another course which I'm involved with the title on Leaking Below Ground Biodiversity and Ecosystem Function in the European forest. So there's a huge interest about what happens below ground and how that affects the plant communities above ground. So we need to go deep down, okay, and see what goes on there. Why it's so important? So the distribution of below ground communities in space and time. They're different in space. If you go in a soil profile, you will find much more organisms at the top soil, at the top few centimeters, and as you go lower, the population changes, but also the types of organisms will change. For example, as for mycorrhizas, even if you go to a coniferous forest, you will see Ectomycorrhiza more in maybe 10 centimeters at the top soil, but as you go deeper in the deeper roots, perhaps in some cases the mycorrhiza association changes and Glomeromycota will occur, okay? Another type of mycorrhiza. So the role of microorganisms in nutrients and carbon cycle, in plant community dynamics, and eco-evolution response to ecosystem and global change. So what happens below ground, I'm repeat that again and again, reflects the situation, how the ecosystem functions because of the nutrient uptake, the carbon cycle. For example, in a conifer forest, if the needles form a leaf litter in the soil, Ectomycorrhiza fungi will be the first who uptake the decomposed nutrient, the nutrients released by the organic substrates or the matter, the organic matter in the leaves or in the trunks and whatever. So what regulates all this association is the soil diversity. When I'm saying soil diversity is the soil conditions, the chemistry, the physical properties of the soil, and that also reflects the function of the terrestrial ecosystem. And these associations are far more important in extreme environments. So we need to define the extreme. What do we mean extremity or extreme? Is it a matter of taste? Is it what I think is extreme for me may not be extreme for you or whatever. But there are some limits, some extremes, or some temperature values or pH values or drought values, which are really limited for all organisms, for all soil organisms, and in return to the plants. So all these limitations, all these extreme situations, they go in pairs. Hot and cold situations, acid and alkaline situations, and salinity and zero or drought conditions. They go together. So bacterial, protestant, mycorrhiza, and faunal taxa in the soil have restricted global distribution due to the variation in climatic, soil, and plant conditions. I'm presenting here the famous triangle situation: the plant, the fungus, and the soil. All three of those are three different environments. Okay, the fungus has a relation to the plant, absorbs the carbohydrates. The plant creates a unique environment for the fungus, releases sugar in the soil, so creates a unique situations, unique environment. Maybe some fungi do not prefer this, or some others they are in favor of these conditions. And some fungi forming mycorrhiza, for example, they grow inside the roots, so the plant itself can control the growth of the fungus in a high extent. Soil conditions, again, as we mentioned before, pH, nutrients, water, humidity in the soil, all these in a combination, they can control the mycorrhiza symbiosis. Again, as we speak about extreme conditions, there is no everything everywhere, okay? Even if you have a global organisms distributed all over the globe, there are some limitations, okay? Some areas which they cannot go. So what's the case for mycorrhizas? Okay? The first among forces, the most famous mycorrhiza type is Ectomycorrhiza symbiosis. Again, this involves the fungus and the plant. From the fungus point of view, is Basidiomycota, Ascomycota, as we mentioned before. And from the plants are not all the families, all the taxa, all the plant taxa who can create this type of symbiosis. The thing there is that there is a huge biodiversity in the fungi forming this symbiosis, and not so high diversity in the plants. So we have a large number of species from the fungi point of view, and not so many plants. Characteristics on these are the type of growth for the roots. I mean, the root architecture, they grow not so extensive branching, but they grow in size, and they become thicker, and also the branching in a unique way depending from the fungus. The fungus forms a cover around the root, the hearting net, and extends the hyphae into the soil, looking for nutrients from inorganic sources or organic sources. Some people very recently, they start to argue if all Ectomycorrhiza fungi are really Ectomycorrhizas. For example, for Lactarius, there is a huge question mark if it's really Ectomycorrhiza, or just by opportunity colonize the plant roots. So we're not really sure. Of course, they create a lot of benefits for the plants, right, in terms of nutrition, but in some species there are huge question marks. Another type of mycorrhiza association, which may be used as a tool in land restoration, is Ericoid mycorrhizas. Again, there is a high specification between the fungus and the plant. These fungi are from the same genera as in Ectomycorrhiza, but they have the ability to form this symbiosis only with Ericaceae plants. Okay? Erica arborea, Manipuliflora, Cistus, and so on. So these plants from this family. This symbiosis is unique. It's very valuable for us, which are dealing with restorations, because they reduce the toxicity created by low pH soils. For example, in high acidic situations, there is a lot of aluminum ions released in the soil, so they create a toxicity to the roots and so on. With this mechanism, with this symbiosis, the hyphae of the fungus can absorb all the aluminum ions and hold them there, so they never reach the plant. So there's no toxicity effect on this. So that's why they form unique environments, these erica plants, the heathlands, and there are certain types of dry heathlands growing in the Mediterranean region, or wet heathlands, as you can see them in Scotland, for example, in Lithuania and these kind of countries. So going back to Glomeromycota, the phylum of Glomeromycota, which I think it's the most important symbiotic fungi phylum, I'd say. Okay. It only has 254 species. They are in all over the globe. They have the ability to form symbiosis with all the plants, so there's no specialization. So, and they are around for at least 600 million years. So there is a question, why you can answer now if you like, if you have any ideas why this is happening. Only 250 species and so long on this planet, right? What do you think? So we can have an interaction. Any ideas? Anyone? Well, actually, these fungi, because they don't have the ability to, they can colonize all the plants. The plant is, yeah.
I have an idea. Maybe we can say this. They have high tolerance. They can adapt with different climatic conditions.
But what happens is that the plant changes itself in order to accommodate to accommodate in a better way this symbiotic fungi. So the plant will change the genome or few changes in order to accommodate better or to increase the efficiency of this symbiosis, and not the fungus. Okay, so that produces a lot of plant species. So some people, they argue that, they describe that this fungi, this symbiosis, is the reason why we have so many plants around the globe. Okay. So how this symbiosis starts? In very old times, geological times, cyanobacteria were one of the most common photosynthetic organisms. So there were some fungi of this phylum able to be colonized by the Cyanoficus. Okay? So the Cyanoficus was inside the fungus and the fungus was around. At that time, that was something like 600 million years, or maybe even before that. The atmosphere of the Earth was very different, okay, in composition. So the fungus could grow on the surface of the Earth without any problem and absorb carbon from the atmosphere because of methane, ethane, and all these nice gases which used to be around. When oxygen came to the atmosphere, that was not anymore possible. So the photosynthetic part went outside and the fungus started to grow inside. The situation as it was described before with the Cyanoficus in relation with Glomeromycota is still around with symbiosis, call it nostoc, in some very wet soils, first described in Germany, and now I think it's all over the globe as well, in very wet soils, in very saturated soils with humid. And as the geological history move on, We have fossil records also suggesting a mycorrhiza symbiosis with pteridophytes, and these are 3 images. And this is the modern days. Okay? These are spores of these fungi of Glomeromycota in modern soils. I want from you to draw the attention that the first 2 at the top are very look-alike, right? But they are separate at least for 400 million years. This is the brown image is from a horsetail plant. I don't know the scientific name is horsetail. Cuisetum. And the other one is from a plantango. But they look the same. Maybe they had the same function as well, but we are not sure. But they look very similar. Okay, the fungus has these spores. They are like small spheres, not all the time, but most of the times in most of the species. And what it happens is they looking for a signal from the plant. If there are some roots around, they will germinate. They will create a hyphae, like the one on the top, on the low right image, and that hyphae will travel to the roots. As soon as they find the root, they will colonize that, and they will perform a structure called appressorium, and then they will try that. Excuse me. They will form a network of hyphae inside the roots, depending from the fungus and the type of the architectural formation of this hyphae inside the roots will be related with the plant and with a fungal species. This appressorium could be created in the root epidermis or in the root hairs, as in the low image. And as the colonization goes on, other structures of these hyphae will be created. These are vesicles. The vesicles are like huge bags full of lipids. And they are inside the roots, or sometimes in some fungal species, you can have spores inside the root. So these vesicles, we said that there are huge containers of lipids. So they store carbohydrates for the difficult times. Okay? The plant doesn't photosynthesize in the same rate all year around. Okay? So the fungus has to eat to gain carbohydrates even in the times where the plant doesn't have a high photosynthetic rate or whatever, okay? During the winter, for example. One of the most characteristic features of this fungi is this abacule. It's like a hand or over-branching hyphae inside the cell, but be careful. Although they penetrate the cell wall, they never penetrate the cell epidermis. Okay, so they never go inside the cell membrane. Okay, so that's the distinguished difference between the pathogenic fungi and this, the Glomeromycota. Okay, so you have an over-branching root membrane around the hyphae, which means that you have a huge area for exchange of nutrients and carbohydrates. These actually are the organs of the fungus which the higher exchange rates of nutrients and carbohydrates occur. Okay? Also, the hyphae, as you can see in these 3 images, they can grow between the cell space, forming the arum type of Arbuscular mycorrhizae symbiosis, or they can grow as penetrating the cells like a kebab, you can see the hyphae, an abacule, and the hyphae continues from the other side. So they can grow and in this way. And this is the Paris type from Paris quadriforme, as it was the first plant described this symbiosis, this architectural pattern. Now the function of this symbiosis. The effect on the plant starts well before the establishment of the symbiosis. Arbuscular mycorrhiza fungi could interact with a plant genome. Actually, they can block the expression of phosphate transporters, or they can reduce their expression in some plants. So you can see, for example, in this graph on the right from, well, there are some references, but you cannot see them here. The blue color indicates that there is no will be the light blue is reduced phosphate absorption expression. The dark blue is absence of phosphate absorbers. Okay, the expression of these genes or related genes with this. So you can see that that thing starts before the colonization, when the fungus approaches the root, and as soon as the mycorrhiza works perfect, for the cells, at least the cells who are colonized by the fungus, there is no, the fungal absorbers are switched off. What it means? That the mycorrhiza becomes the main pathway, at least for phosphate. Okay? The plant always keeps both ways on absorbing phosphate, but in soils with low availability of phosphate, prefers to use the mycorrhiza symbiosis. If the phosphate is much more available in the soil, the plant absorbs this phosphate from the soil by itself, by the root hairs or the root itself. Why? It's a matter of energy. The plant in symbiosis and having the need for phosphate in a low availability phosphate soils has to spend a lot of energy to absorb the phosphate from the soil. I have to remind you that the phosphate ions in the soil, they cannot move around like many other ions. They stay there. So the plant roots have to travel to the phosphate. Okay? So that means that if there are limited source of phosphate in the soil, the plant has to produce a lot of roots. So that is a high energy cost function. Okay? That's why they prefer to form mycorrhiza. We'll see later on in an explanation on this. Similar is the situation for nitrogen. Although for Glomeromycota, there's no specific pathway explaining the nitrogen increase in nutrition. Probably there is a relation with bacteria which are growing around the hyphae in the soil, in the rhizospheric soil. So, well, that's my opinion as well, that these bacteria absorb the nitrogen from the soil and they move it, they can help the fungal hyphae to absorb the nitrogen from the soil and transport it to the plant. So we describe so far an increase on plant nutrition as a matter of phosphate, and of course similar mechanisms exist for other nutrients, for potassium, for calcium and zinc and whatever. Other changes which could be very useful for restoration, for land restoration projects and so on. Mycorrhiza symbiosis with a Glomeromycota could increase the root branching, the root biomass. The plant start to produce more roots. For example, in this graph, which is the root, the specific root length, which is centimeters of root per gram, you can see that after different inoculation with this fungi, we have more and more roots. What is very important in this graph is that the root growth follows a pattern like so every something like 10 days we have more roots produced. What it happens is that the very fine roots, they die very quickly when they are in symbiosis. The very, very fine, I mean fourth order roots or fifth order roots if they exist. And then they die, and then the plant produce more and more and more. But that doesn't have any cost impact to the plants. It's just the branching more. Another effect is that we have observed that there is a reduction in the root hairs. The plant doesn't need anymore the root hairs as long as use the pathway from mycorrhiza, and because the root hairs are the main organ absorbing the phosphate, so there is a huge reduction in the root hairs. Okay? That, of course, differs from fungi to fungi and among different plant species, but is a general pattern. Why this happen? Well, there is a new described phytohormone, strigolacton. Strigolacton is expressed by the plant and produce more branches in the more leaves, more branches. Okay, if the P level, the phosphate level in the soil are high. If it's not so high, the phosphate absorption from the plant is not so high, strigolacton has an effect to the plant, reducing the branches, reducing the leaves, but that increases the root branching, and also is the main signal, the plant signal to this soil fungi. So these soil fungi, in order to colonize the plants, they need the signal from the strigolacton, which is much higher in low peel and in low phosphate soils. So that could explain a lot. We said that the phosphate pathway works better in low phosphate soils, and that signal, that phytohormonic signal, could explain why. So all that is a little bit looks like a model, as everything we said up to now. So does mycorrhiza symbiosis works in the extreme? Because we are talking about extreme environments. So some examples of biomes under extreme conditions. For example, the top left is tundra, Arctic conifer forests, grasslands, shrubs, and deserts. Okay? All these plants, they form mycorrhizas. Okay? In man-made environments, okay, or in environments under huge pressure from the humans, from human activities. There are several examples of tallings. The one in the top left, the three top left are from India. The lower middle is from Libyada in Chalkidiki, and well, the three middle ones are from Libyada, and the three on the right are from the same site, the same areas. What it happens there are some, as we as you've heard yesterday, there are some plants having the ability to compensate the toxicity levels of heavy metals, of high pH levels, high or low pH levels. So these plants, not all of them, but some of them, increasing these abilities due to the mycorrhiza symbiosis. These are unique environments. They are very disturbed, very in every centimeter you have very different conditions, horizontally and vertically. Okay, and also you have huge problems with the water. So Arbuscular mycorrhizas and heavy metals. How this works with these plants. Several plant species have the ability, as you heard yesterday, to accumulate large amounts of heavy metal in their tissues. These are Asteraceae, Brassicaceae, Caryophyllaceae, Kiperaceae. For example, Brassicaceae, you had from Professor Tsiripidis several examples of Brassicaceae species which can accumulate high nickel concentrations. Normally, Brassicaceae are the only plant family from the gymnosperm, angiosperm taxa, which they don't form Glomeromycota symbiosis, Arbuscular mycorrhiza symbiosis, because they have a special genome. But these hyperaccumulations from the Brassicaceae family are the only Brassicaceae which they form symbiosis with Glomeromycota. So it's very interesting. So when these plants are tolerant to heavy metal stress, they intensify their symbiosis, higher root colonization with Arbuscular mycorrhiza at the stage of high nutrient demand. So when the plant grows in a heavy metal substrate, they have higher colonization, higher symbiosis with more symbionts with these fungi. So the alleviating potential of Arbuscular mycorrhiza on heavy metal stress is determined by different factors, type and concentration of heavy metal, plant specification, and of course growth conditions. So as you heard again yesterday, in some cases, the inside the hyphae of the fungus, the fungus creates an organometallic molecules which cannot be diluted in the soil. So the metals are not anymore available for the plants, or they just stay inside the root hyphae. So they are excluded from the system. Okay? And also molecular analysis have indicated that mechanisms involved in heavy metal tolerance of the plant. So there's a huge, there are a lot of findings suggesting that some genes from the plants are overexpressed in order to increase the compensation on heavy metal tolerance, okay, from the plants. So this is an example, from Riviera Becerri. The plant was Pisum sativum. They colonized those plants with Arbuscular mycorrhiza, and although the biomass of these plants was decreased due to the heavy metal content in the soil, the mycorrhizic plants were less, they had lower effect on the toxicity effects of, from the heavy metals. when they measure that on the molecular levels, some proteins were more expressed in mycorrhizatic plants. For example, the white bars are control plants. The right next to it is the gray, the light gray is plants inoculated with Zygospora, one fungal species. And these are the controls. The next one is controlled with cadmium. And the next one is, the next bar is Zygospora with metal content in the soil. So you can see the expression of these proteins that is increased under symbiosis, okay, or decreased depending from the role of these phytohormones, okay, or. So this is an experiment performed in Olympiada, where nerium oleander plants were inoculated with a Glomeromycota, and we didn't know what we are going to find, and they work as metal accumulators, specifically in the root system. But again, here the iron, the manganese and copper, not so much for copper, they were trapped in the root, inside the roots, and more specifically inside the hyphae, depending, of course, from the treatment, the soil treatment. Okay, but all the mycorrhizatic plants, they had more metals in the roots. So what do we know so far? That under a heavy metal stress, the unfavorable oxidative effects influence the plant growth. Okay, so when you have a metal stress, there is an oxidative effect in the plant physiology, and that reduces the growth from the plant to the plant. AMF Arbuscular mycorrhizae are able to enhance production of antioxidant enzymes, which can alleviate the stress of these heavy metals. So they reduce the effect of oxidation inside the plant. Arbuscular mycorrhizae via phytostabilization mobilize metals from the soil. These are absorbed by the hyphae and become mobilized there. Okay. But in some cases, not everywhere, these metals remain in the hyphae, so they never go up to the shoots. But there are some other cases which we have increased in the plant tissue, in the above ground plant tissue. And finally, the plants and Arbuscular mycorrhizas, they form exudates in the soil, which they can trap the metal ions and produce immobilized organic substrates, some molecules. Any questions so far?
Do we know why hyperaccumulators grow symbiosis with
Glomeromycota. Well, the first sentence, I think, is the answer. Well, yeah, because normally the plants under stress and under heavy metal stress, they form oxidative effect in the physiology, in their cycle. Okay. With Arbuscular mycorrhizae, they reduce this. Okay, so hyperaccumulators, well, we know now that in some cases, at least, in some plants, they reduce that dramatically. Okay, the effect of mycorrhizal symbiosis is on that level very increased.
Is there a ratio of the absorbed petals between the root system and the shoot? And is this ratio stable or can change due to something? I don't know.
It could be different in different plants in different fungi. Okay.
So it's species dependent for the plant and also for the mycorrhiza species.
Yeah. Okay. So the trick is to find the right plant and the right combination of fungi, because normally one plant doesn't form symbiosis with one fungi. Okay. So it has a group of fungi growing in the root. So you have to find these combinations.
Yeah. Otherwise, in the restoration project, you have to collect the roots also.
Yeah. But in the roots, they are less available. Okay, as long as they remain there, there is a dilution effect, for example. Okay, so you reduce the concentration of the metals after many generations of plants. Okay. Others?
You're probably going to say it, but what are the factors that could degrade the fungi? Are they, for example, soil moisture or factors that could prevent the mycorrhiza from happening, the association from happening?
The only factor that is well described, but it's high phosphate content in the soil.
That's the only one.
They can survive even a high dose of benomyl, for example. Okay. So they can withstand chemical warfare against them, and they exist in a very low pH and a very high pH as well. And they also, well, newly described some species, they can exist in a very high phosphate level as well. So, but in these cases, the plant is not mycorrhiza dependent. Okay. And just to go back to strigolactone expression or strigolactone phytohormones, under salinity stress, the strigolactone is also overproduced. That means the plants produce more strigolactone under salinity, under high salinity, which means more mycorrhiza will come to the plants to compensate this harsh environmental situation. Okay. Again, the graph is from the same paper. So Arbuscular, just to summarize this, Arbuscular mycorrhiza plants increase strigolactone production in order to promote symbiosis and establishment to cope with soil stress. So what I think it's happened is with heavy metals and salinity stress, the plant behaves more or less in a similar, behaves similar. So the symbionts interact with the plant, make it demanding or making the plant actually to produce more strigolactone, so they become more mycorrhiza dependent. Okay, strigolactone is the phytohormone responsible for mycorrhiza symbiosis. Arbuscular mycorrhiza and water stress. Again, in extreme environments, water stress is a key, a significant stress. In the Mediterranean areas, we've heard so many lectures about water stress today and yesterday. So plants under water stress could show an increased acetylene oxidase reaction. This reaction is an expression of a stress, and the plants become more hard, and they don't grow. They become dry or less green after a while. Plants in Arbuscular mycorrhizal symbiosis have been shown to alleviate this oxidate damage and have lower lignification under drought conditions where hyperoxide cycle was significantly higher when Arbuscular mycorrhiza was not present in the system. Okay, what that means? That means that the lignification, okay, in the grass in the fields occurs a bit later on with mycorrhizic plants. So they stay green more. So because we had the lectures today for related with goats, sheep, and animals feeding from the grasses. So presumably, well, when there's extensive mycorrhizal symbiosis in the system, the animals could be fed by the plants for more time. Okay? And of course, another example with mycorrhizic plants and drought conditions. This is a different population of Glomeromycota. There's no control plants in this graph, so there's no artificial, for example, plants. All of them have mycorrhizas, and you can see that the expression of related effects from the drought are different among different mycorrhiza fungi. And again, another effect on the physiology of the plant. Catalan superoxidase is more expressed in some mycorrhizic plants. Again, when you choose to interfere with the natural Glomeromycota population and to introduce Glomeromycota into the system, you have to be careful to not to reduce these beneficial effects from the plants. We have some lectures today that we say that some plants were adapted to dry conditions, for example, in some ecosystems. Okay, so probably that is a reflection into the soil. Okay, because these plant populations are colonized by these fungi who alter these abilities to these local indigenous grasses, for example. Okay, and as we observe with in a paper with Apostles and Eleni, the number of tillers in Dactylis glomerata after inoculation with different Arbuscular mycorrhiza change after different drought treatments. So again, we observe the same effect, that the plants remain more nutritious, maybe, able to feed the animals for longer periods after mycorrhization. Okay, because also we had a different response, an increased response in the number of the leaves and tillers. Okay. Again, one of the main all these effects were in the plant itself. Okay, but one of the major impacts of Glomeromycota to the soils are the formation of microaggregates. Microaggregates are formed mostly by the protein released by these fungi to the soil, glomalin. And glomalin is strong hydrophobic protein, which means just around this a bubble of glomalin, all these soil particles will start to be tracked, okay, to form microaggregates. These microaggregates are an environment for bacteria. Organic matter trapped inside these microaggregates could stay there for much longer periods because it will decompose much more slower. And these microaggregates are the first building blocks of an actual soil, okay? Because a normal soil should have aggregation. Okay? When we go to restore a site, one of the main goals is to produce physical properties for the soil, to produce aggregation. Okay? So I was a bit faster than the previous. So what we have learned, all right, in this talk, how the these will be the your questions. I mean, the questions for you. We can discuss now some of them. How the mycorrhizas could alter the plant response to environmental stress. If mycorrhiza fungi exist in almost every soil, why do we need to apply them artificially? Okay, probably you have heard that there are some mycorrhiza products in the market, so you can go and buy them instead of fertilizers, for example. So why do we have to apply them in the first place? Okay, and when we have degraded sites, do we leave them as they are and hoping that they will restore by themselves, or we intervene with restoration projects, and why we have the need for that? Okay, we can start to discuss that with the first question, for example. Any ideas from you?
Sometimes maybe we can use the fungi as a catalyst, for example, because sometimes some action will take place and need a push. You can use this as a first step to prepare best situation to plant come and grow.
Is that for the second question?
Yes.
Yeah, okay. Yeah, but the local fungi will be much more adapted to the soil conditions there, okay? So, yeah?
Also, for some degraded soils, we need to make artificial inoculum fungi to restore soils.
Yeah, why? Why?
Because
Are the degraded soils have mycorrhiza fungi or not? Because in most of the cases, okay, apart from very extremes, in tilings, for example, in ores, okay, there are few plants growing there, okay?
Yeah.
Even in tilings, you will find tamarics, for example. But do they have mycorrhizas these there or not?
they have, but
So why you not isolate the fungi from there and apply these fungi to the plants? Instead of apply a commercial product, for example. Commercial product, most of the times have 5 or 10 fungi from all over the globe or many sites from the globe, okay? and it's certified that produce better plants. Okay.
Because we increase the number of traits, the diversity of the fungi, so we have more functions basically to help the plant to grow.
I think you've heard it in the previous lecture. The first year is the most important, all right, for a successful restoration project. So you need to enhance the growth in the very first months or years, okay, of the project. So if the plants are not successful in these first moments, okay, then you fail. Simple as that, okay.
And also, they play important role in soil formation also. And in this case, can help the plant.
That's the next step.
Because
In remediation, you have to create soil, normal soil.
yes,
That will take years, okay, in most of the cases. 5 or 7 or maybe more, depends from the site and the conditions. So the existence of mycorrhizas, first you restore the below ground functions, okay, biological functions, bacteria, fungi, all these, okay. That's why I said that in the first or second slide that below ground biodiversity is a key for a successful ecosystem function.
and soil formation and after that soil development.
So do we need to remediate the sites, or should we leave them in their own fate? Because if you leave them like that, eventually metallophytes, for example, will colonize the site, or plants who normally exist in these extreme conditions, they will colonize this site. So what do? You intervene or not, and why?
So it depends on your objective. If in some areas very close to the community, people expect a faster recolonization, so you have to intervene with this.
If humans are living next to mine sites, for example, then you have to intervene with the site quite fast,
Yeah.
because there is a huge problem of pollution, but is this always the case, or you have to leave them as they are if it's in a remote areas or whatever?
So maybe also depend on the distance of seed bank. Because it's new created this soil, so maybe there's no local species can adopt to this kind of place. Maybe it's far from the seed bank.
So
it's not easy to do for them to reach this area.
Well, what we did in the lab, okay, in my lab, or the lab which I used to be, we intervene in any case, okay. We try to enhance the process. We introduce metallophytes, for example, plants from the normally from the surrounding areas of the site which are going to be remediate or rehabilitate or whatever, okay. But also introduce similar plants from other sites there, okay. Just because we need to stabilize first the site, to start create soil. The first step is to create underground functions, okay, inside the substrate, fungi, bacteria, and all this, because they are very reduced, because it's high disturbed environment, especially tallins or mineral extraction points, or all the factory sites and industrial sites, Okay. And the second step is to try to enhance the process of creating soil and organic matter. Okay? That's the two key steps. And you should always, well, that's our strategy anyway, you should always take these two steps as soon as possible. Okay? After that, you can wait more and more years.
Is there no way to spread the fungi directly in the soil instead?
Yeah, yeah. Because they come as in beads or in powder.
Oh, okay.
So you can. It's very easy to distribute them with irrigation water. With this, in the last talk we had that hose with like a fiber. In Israel, they use that quite extensively. They introduce Glomeromycota in the irrigation water, and through these pipes, the spores are spread to the field. So there's an inoculation with this in this way.
So it's also called inoculation.
I think it's something to consider when you collect seeds, to think of
Yeah, normally
some soil and to inoculate your seeds in the greenhouse.
yesterday it was a talk from you
If
in
you
Jordan.
cannot
Was Jordan or Palestine in Gaza Strip?
If you cannot spread the soil because the area is too big.
In that project, they collect the first centimeters of soil. With this, they collect the seeds, the seed bank, and also the Glomeromycota existing in the area. Okay.
Sometimes you cannot restore the land is too big. You cannot cover it all over. So you can think of inoculate.
No, these are fast growing fungi. I mean, in a matter of days, they can colonize the whole room, for example. Okay. So even with a few inoculation points in this room, in a day or two, you will have these fungi all over the place. So they fast growing, and so as long as you have living plants inside the room, for example, then the fungi will spread very rapidly.
The question is, when do I need to inoculate them artificially? Because I understand when you have mineral soil or.
As I said, if there's a high demand on restoration, because there's a risk of pollution, for example, or the dust produced by abandoning the site is toxic, for example, to metals or whatever, zinc, for example. Then you have to create a cover, a grass cover or plant cover of the site. So you need to enhance the process. Then you have to apply this kind of fungi quite rapidly. Okay?
Yeah, just a suggestion. If I take Alejandro's plant material and bring it to the area, then I do an inoculation of mycorrhiza anyway, or?
Yeah.
Because they exist in the nursery.
Depends on the soil that you're using.
Normally in the nurseries, the plants may have one or two fungi with them, but in some cases, when they use acidic turf to germinate the seeds, then there is no mycorrhiza, because they apply a lot of phosphate, and the turf is very, very good substrate, so the plant doesn't need the mycorrhiza symbiosis, so doesn't create this. And that sometimes in the field, well, I found that as well in some studies. When you have the plant from a nursery without mycorrhiza, the transplanting stress is much higher because the roots are growing in such a way, so when they placed in a harsh environment, the natural environment, away from the nursery, okay, they die The roots are dying. So that has an effect on the plant. If the roots are inoculated with mycorrhiza, they are much more branched than the after phosphate application. And even if they die, the very fine roots are dying, okay? Which is not significant cost to the plant. So the plant keeps going on.
Do they cost a lot, the commercially available?
No.
They're cheap.
In the past decade is a huge development on that. If you ask me the same question something like 10 years ago, I would say that it's very, very expensive. Nowadays it's not so expensive. If you don't have any other questions?
Mhm.